hopefully talking about tertiary structure in just a few minutes, but we can kind of, if I don't finish all of these, hopefully having done the exercise, you don't need me to come back and finish them. We'll see if there are any slides that you need to see again. So we ended last Friday with secondary structure elements, alpha helices, beta sheets, and that some proteins are all one or all the other. But that's really the exception. Most proteins are combinations of secondary structure elements, and not just alpha helices and beta sheets, but what do you call the parts of the protein that are neither alpha helical nor beta sheet? So in textbooks, you would often see, so instead of this coiled region, sometimes people used to call them random coil. And that kind of implies it adopts one structure when it feels like it and another structure when it feels like it. So that's not true, which is why random coil is not a great term, but people still use it. So it just means it's hard to describe, or it doesn't fit into the alpha helical or beta sheet category. Okay, so random coil or coiled region just means the backbone does something that's not a regular secondary structure element. Okay, and of course, if you're going from one element to another, or especially if you have an anti-parallel beta sheet, so you have to get back to where you're going, right? So if the polypeptide backbone is going in one direction and then it has to change direction, somehow you have to connect those two strands. So those changes in direction are what are called turns. And I don't care that you learn whether it's type 1 or type 2, we're just going to call these beta turns and just say there are two main ways that they can form, okay? And so what I didn't tell you when we were talking about peptide bonds is that because the peptide bond has a partial double bond character, we can think of whether it's cis or trans, right? So if something has, a double bond, are the R groups on the same side or on opposite sides? So are they cis or are they trans? So what would you guess for a peptide bond, which again is not fully secondary, not fully double bonded? Would you guess that the R groups are on the same side or the opposite side? Are they more likely to be cis or trans? Trans, just for steric reasons. You got this big old thing hanging off, have a peptide bond, the next amino acid, the big old thing should be going in the other direction. So 99.9 whatever % of the time they're going to be trans. So the exception to that is when you have a proline. Okay, so even if one of the R groups is proline, it's still maybe 99.8% trans, but occasionally you'll have a cis peptide bond if the next amino acid is a proline. Does that make sense? Especially if that proline is in a turn. Okay, so this is one of the kinds of polypeptide beta turns that can be made. So if you have a proline, it kind of forces the backbone to make that turn. So a cis proline is often found in turns of polypeptides. Okay, so we're calling these beta turns. So one way to make a nice tight turn in a polypeptide strand is if you have a cis proline. The other way to make a nice tight turn is having a glycine. Okay, so again, glycine, no real side chain of significance, and so you have less steric hindrance overall, and so it's very easy to make a very tight turn of the polypeptide backbone. You don't need to learn that one's type 1, one's type 2. Just know that those are the two kind of unusual or exceptional amino acids that are often found in polypeptide turns. Okay, and so what do we have here and here? Hydrogen bonding. Okay, so this is not the hydrogen bond that's holding an alpha helix together or a beta sheet together, but it's as those backbones come close together, might as well make a hydrogen bond that'll stabilize it even more. Okay, so that tight turn, either of a cis-proline or a glycine in particular, allows that tight turn and the extra hydrogen bond for more stability. So we talked about primary structure or sequence, secondary structure elements, connecting the secondary structure elements, and then you have what more people would call motifs. So we could call them supersecondary structure elements or structural motifs. So patterns of secondary structure pieces that show up again and again in proteins. Okay, and so they're often named in obvious ways. So beta, alpha, alpha corner. I don't know that I've ever seen anyone mention an alpha corner, but that's kind of what it looks like. So you'd have these kind of repeated units that are just used over and over again as a convenient way to connect the polypeptide backbone. Okay, so now we're going to get to tertiary structure. So we're going to build up those secondary structure elements with turns and connections into tertiary structure. So I would say, how would we define tertiary structure? Is the fully folded polypeptide. Okay, and so some people would call the tertiary structure the compact structure. So if we think about the primary structure as all the way extended, now secondary structure we have little pieces that fold up locally, or a piece that kind of comes back around in the case of a beta sheet. Now tertiary structure is folded properly and it's all one, typically polypeptide. Okay, and so again, there's our alpha helix on the left, then the beta sheet. Now we put the pieces together and we have our compact, fully folded polypeptide. So that's our tertiary structure. And so the structure on the right is actually a drawing, so a reproduction of a drawing by this great woman at Duke named Jane Richardson. Okay, so Jane Richardson is probably in her 70s, maybe pushing 80, and she is one of the cool stories of women in chemistry, women in science. So as if we haven't lectured little enough today, I will give you a little side story. So Jane Richardson is a crystallographer. She and her husband, I think David Richardson, so he's a crystallographer, one of the early crystallographers. They have deposited some crystal structures into the protein database, some of the very earliest structures, so in the 1960s and 70s. They're not young. And at the time, so do you think we had a lot of great computer graphics capabilities in the 60s, 70s, 80s? No. So how would she represent, on behalf of her husband, how would she represent these structures? She would draw them by hand. Okay, so now we have very sophisticated three-dimensional viewing programs. She would draw them, and so she is the person who came up with the idea of a cylinder to represent an alpha helix and an arrow to represent a beta sheet. And so because her drawings are artistic but also visually helpful for understanding proteins, people would collect her drawings as artwork and hang it on their walls. So if you have an original Jane Richardson, that's a big deal. It's almost like Andy Warhol. And she's still alive. She's still working. Well, they both are. She does not have a PhD, but she's a fully tenured faculty member at Duke in the chemistry department. On committees, advises grad students, because her husband got a PhD. She married him. She followed him. Duke hired him. Often in that era, kind of women hung out in their husbands' labs, helped, worked side by side, never bothered getting their PhDs, but she is at least as smart and just as capable and a valued member of the chemistry department at Duke, and a great lady. Who wears her hair in this big old braided bun on top of her head and probably is all the way down to here, I don't know. So in addition to primary, secondary, super secondary structure, we also have, and I guess I would call this, well, so here's where semantics come into play. So this is a picture of a zinc finger motif. Okay, and so even though it's not the whole protein, it represents a tertiary interaction. So I guess, so this zinc finger should probably be on the super secondary slide because it's not the whole polypeptide. It brings together secondary structure elements. So what secondary structure elements do you see here in this image? So there's a little bit of alpha helix, a little bit of beta sheet, and now this is your first metalloprotein you've seen. So the zinc ion is being held in place by what amino acids? So what are the cyan ones? So those are histidines, and the orange ones are cysteines. You probably can't tell that from looking at it. So 2 cysteine, 2 histidine, chelating that zinc in place, and so that's bringing parts of the protein that are a little further away in sequence close in space. So it's contributing to the tertiary folding of the protein, but by itself it's just a super secondary structure element. So that's where it's kind of bridging the gap between a motif and tertiary structure. But when we talk about tertiary structure, what drives that? So it's really the hydrophobic packing of a protein, getting the amino acids away from water. So it's dominated by hydrophobic interactions. Electrostatic, hydrogen bonding, not such a big deal. In tertiary structure, that's where we also see disulfide bonds. Okay, so remember disulfide bonds are just oxidized cysteine residues that come close in space, and often metal ions will help assemble tertiary structure. Okay, so now we assemble these little pieces, either super-secondary structure motifs or secondary structure elements, into the overall protein, and you can kind of see that little piece extracted from the structure. Okay, and so let's just talk about the hydrophobic effect for nobody did 2 minutes. Okay, well, we had fun anyway, right? Okay, look over those. I will ask you if you have questions. We'll talk about the hydrophobic effects, but there may be stuff on there that we won't directly cover. We'll see.